All right, let's go to a little bit of labs. I'll probably speed it up a little bit. So for labs, the list is looking at shifts to high and low, looking at scatter plots, looking at box plots, and shift tables, right? So I don't know what people's normal practices are, but I'd say labs, there's even more variation than adverse events, right? We have plenty of variation in adverse events. We probably have even more so with labs. Okay, so but even though there's other recommendations, the idea is they wouldn't necessarily all need to go in the CSR, right? Some databases could be created if you are lucky enough to have an interactive tool internally, right? You can do some of these things with your interactive tool, and if there's something meaningful or something worth sharing in a CSR, well, then you can put it in the CSR. So just because this package, it seems a little big for labs, I mean, it wouldn't necessarily all go in a CSR, but this is the package we would use for signal detection. So some common pitfalls is creating summaries for all labs in a data set, right? It's really easy for a programmer to run their program in every single lab that just happens to be sitting in SDTM or ADAM, right? That's something that probably needs to stop. What usually happens is that does lead to some, a lot of volume that you may not need, and some of those labs may have been there for just individual patient care purposes or case review purposes. Some of them don't really mean a lot as a group level summary, so you want to be smart on what labs you include in your group level summary. In particular, there's these labs collected in a reflex manner. What that means, you only collect a lab if something else happened, right? So if you saw a value greater than something, then I'm going to collect this other lab. Well, we call that a reflex manner, and doing a group level summary on those labs adds no value. I mean, it's basically a biased group of people who had something done, and it could be just the bad results, and how is that going to help you with signal detection? That's a common pitfall. And we also get some other labs that microscopic exam or cell morphology, sometimes there are labs that the only thing you get are positive results, right? You only get things if they see something. And those, you can't just put them through a statistical program and get anything meaningful out of that, right? Those are you're gonna end up with 100% of abnormalities. You might get a denominator of 2 and a numerator of 2. It's like that's when you know something. Usually, when you see a table with 2 out of 2, that means you probably analyzed a lab you shouldn't have been analyzing, right? It meant it was probably a lab that you only get the positives for. These are all very common pitfalls that we need to look for. Okay, this is the shell. It's in the white paper, but it does count their little n, big N % of people who shift to high or low. This is the scatter plot that we like. We are trying to definitely get to more figures when it comes to lab and vital sign data. So the scatter plot looks at your baseline versus your max post-baseline if you're interested in high values, or min post-baseline if you're interested in low values. And this is a really nice way to find any patients of interest. It does add to your treatment emergent high-low, because your table, your little n big N % table, will never tell you how high are your highs getting or how low are your lows getting, right? And that's a little frustrating for someone who's trying to interpret this data. This will at least tell you how high is high, how low is low, and it tells you where you are with these labs. We also tend to like box plots. It seems to show changes over time very well, so it's a recommended display. This shift table, which is low normal high at baseline and low normal high post-baseline, this is very popular. A lot of people like this. If you read our white paper, it says that we don't feel this adds a whole lot of value over the other things, already having the box plot, the scatter plot, shift to high, shift to low in little n big N % format. This one is very popular. We kind of keep it in the recommendations only because of that, but we hope over time, as people get used to scatter plots or spaghetti plots or these other plots, maybe someday we can get rid of this. We only recommend it because we're scared if we didn't recommend it, you will be almost certainly be asked for it, right? We're just worried that you would almost certainly be asked for it. But hopefully this will eventually move away from our recommendations, mostly because it's just a whole lot of data that's hard to look at, right? Visuals are better. Like we have better visuals for looking at this kind of data. Okay, that's shift to high for during studies, during study and after. So the same rationale as in adverse events, right? The whole package is built for signal detection purposes, right? We're trying to look for imbalances between drug and placebo, or in the uncontrolled area, we're trying to look for patients, individual patients who might need to be looked at individually for medical. So those are our main purposes, which is why we like some of the scatter plots, and we like the looking at comparisons of percentages of people who shift. All right, so I think I will briefly go over this. So the shift to high-low is a nice way to look for that imbalance, right? Because you have percentages. You can look for an imbalance between treatment and control. You can look at magnitude of effect. So that's where that table fits in. The scatter plot fits in on trying to look at how high is high, how low is low. The box plots will look to see. It's a little bit more data because it's over time, and also only recommended for when you have controlled data. It's not as useful when you have uncontrolled data, but it looks for any kind of separation with treatment. And I already said the shift table is not considered useful, but maybe we can get rid of it. And a lot of people do change from baseline to endpoint. That's a very popular analysis for lab. It's also currently in the recommendations only because if you don't do it, we're scared that somebody will make you do it, and we're trying to be realistic, right? So this, even though it seems odd, we recommend something we don't really think has a purpose, but we're also trying to put on a realistic hat, and we don't want somebody submitting a CSR only to get asked for something more. This is something we do hope to phase out too. A change to last observation has no purpose in signal detection, right? Because if your drug causes a change in a lab, and let's say it happens right away, and maybe you go back down to normal right away, you have that blip, well, guess what? That could still be an adverse drug reaction. Just because it went away by the end, yeah, it's important to know, but now you're starting to get into characterization. It might be transient, right? But you still need to know if your drug causes something, even if it was transient. That still needs to be in the drug's label. Right, so if you look at change from last observation, that really has little value in signal detection. It's also not good for signal characterization either, right? It's actually pretty poor for that too, and I'll show you an example eventually. I think it's a later slide. All right, so one frequently asked question is, why do we have so many displays for labs? So most of them do have a purpose, but as I mentioned, a couple of them don't. And we just need to try to phase them out of predominant practice, but we're kind of in predominant practice for a little bit, but hopefully we do get phased out. Another reminder is that there is this FDA guidance on late stage, FDA's 2016 late stage guidance. As your compound matures, right, you should stop collecting most of these labs, right? And so that's something that I'm not sure. I think people are shy to do that, or companies are shy to stop collecting labs. Well, there's a whole guidance document that basically says it's you probably should do that. There's just to, spend our research dollars wisely, right? Eventually you can stop collecting labs if you have a mature compound. Okay, I'm going to skip that one. All right, so one frequently asked question is, well, we use MMRM, right, mixed modeling, for efficacy. Why wouldn't we do that for labs? I don't know if anybody gets that question. We get that question sometimes. But the main idea is, for efficacy, you're usually trying to estimate different things than you are for safety, right? So just because the data structure might be similar doesn't mean you should use the same analytical technique, right? You might be estimating different things. And efficacy is usually estimating kind of an endpoint type summary, and as I mentioned, endpoint for safety is not as useful, right? We still need to be concerned about things that happen in between. So that is not something that's generally, there could be some adverse events of special interest where this makes sense, but for your run-of-the-mill lab, doesn't seem to have a big purpose. All right, so another thing that comes up for labs and analytical planning is what to do with your unplanned or unscheduled labs. We do recommend that they are included when we look at shifts to high, shifts to low, but when we look at box plots or any changes over time, we recommend they are excluded. So why would we exclude those from a box plot over time? Well, if your main purpose is looking to see how your mean changes over time, having an unscheduled at a visit, having most of your boxes be on 100 patients, and you only have a box on one patient, that really screws up your interpretation, right? It doesn't really help. So we're thinking because those are covered in other analyses, we feel very comfortable that they're excluded from those types of summaries. All right, so another thing that comes up with labs is what units to use, right? So there's a 2013 FDA letter that basically says, we're sorry, we're not used to SI units yet. Please give us stuff in CN units, right? In reality, what that means for us is we're often doubling our analyses. We have we have to have a whole set in SI units, a whole other set in conventional units. But some of our recommended displays, if you read the white papers, we are trying to get creative about that, right? So a scatter plot can have both SI and CN in the legend of the plot, right? You don't have to double your work. Same with a box plot, right? So some of these things, we have creative ways, and that's part of why we want to move together as an industry, dealing with some of these things similarly and learn from each other, right? So we do have ways we can try to handle both units more creatively than just doubling our work. All right, so I briefly mentioned about looking at group means. This is a very common pitfall. Using group means to determine whether or not your lab changes are persistent or transient. People like to look at group means, often it's box plot or a line chart, or an ugly table. If you have group means over time, it's really unreliable, right, to use that data to say whether or not it's persistent or transient. And here's our example. So this is a very extreme example just to get a point across. So let's say your placebo patients just kind of have normal variation, not too many people changing. Okay, but your drug group, let's say you have a small percentage of your drug people who have a lab that goes way high, just goes out of whack. So you have a small group that goes way high. And then when they go way high, they stop. They discontinue drug. They either by protocol or by investigator decision or whatever. But some protocols say if you have certain labs at a certain level, you need to discontinue. So it doesn't matter how you do your group means, right? If you do observed changes over time, it's going to look like it's transient. If you do last observation carried forward over time, it's going to look like it's persistent. If you do MMRM, it's going to look like it's transient, right? Guess what the real answer is? The real answer is you don't know. Everybody who had that increase didn't stay in the study. You don't know if it's persistent or transient, right? So it's very easy to fall in this trap to use these group means and try to determine whether something is transient or not. We do so this is a case where you just might need to look at individual patient data, the spaghetti plots, right? So if you just looking at all the lines just to know to make sure you're not falling into this trap. This is a very common pitfall, even among statisticians, right? Let alone medical people.